Um, it's time. It's time to start. Hi, everyone. My name is Olga Tarkhanova. I'm a pharmaceutical chemist. My PhD in pharmaceutical science, and since 2019, I work at Chemspace. And 2 years ago, I was assigned to be CEO. I'm extremely happy to host this webinar today. And before we start, there are a few rules. First of all the participants are on mute. If you have some questions or comments, you can type them in the chat box. This webinar will be recorded, and the recording will be accessible on our YouTube channel. So everyone who, for some reason, missed this webinar can access it on our YouTube channel. So today's agenda, I will start with introducing Chemspace Integrated Discovery Platform. It will take a few minutes max, and then we will come to the actual topic of our webinar, and you're going to learn about V-SYNTHES approach, how it works, and also recent expansions and application of this approach. And finally, we're going to get Q&A session. So you can type your questions in the chat box, and then I will read them, and you will have your questions answered. I'm very proud and extremely happy to introduce our speaker today. This is Professor Vsevolod Katritch from University of Southern California. He's professor of quantitative and computational biology, chemistry, and pharmacology, and he also is a co-director in the Center of the New Technologies in the Drug Discovery and Development at the University of Southern California. Seva is a computational biologist and a computational chemist. His main focus is in studying molecular function and structural pharmacology of membrane protein, primarily GPCR proteins. He develops and applies new physics-based and AI-based technologies to facilitate discovery of new ligand and potential drugs for GPCRs and other targets. He did a lot in the direction of chemical space exploration. His work has led to 9 patents and more than 150 papers. He is named among highly cited researchers by Web of Science in 2 categories, pharmacology, toxicology, and biology biochemistry. Seva and his lab, they developed this V-SYNTHES approach, which helps us to explore extremely large chemical spaces over trillions of molecules. And we are very proud to have this technology, to have V-SYNTHES technology in our portfolio and utilize this technology for hit identification within drug discovery process. We have been collaborating with Seva for more than 5 years, I believe, and it was, of course, always successful collaboration, but also very joyful, I would say. And I really hope that we will continue work together in the future. Seva is our scientific advisor, and we always get very valuable advices with our projects, with our tasks. A few words about the integrated discovery platform, which we enabled a few years ago. This platform built on the capabilities and facilities of 2 companies, Chemspace and Inami. We're present globally. Our headquarters is located in Kyiv, Ukraine, and our extensive expertise in integrated drug discovery allowed us successfully accomplish more than 100 projects related to drug discovery. We offer custom solutions, and we have a number of satisfied customers across the world. The integrated discovery platform consists of different components like protein science, biology, chemistry, pharmacology, computational science, molecular modeling, and screening. So what we do, we tailor these components rationally to accomplish each stage of drug discovery process rapidly and efficiently. What is also important to mention that our focus is always on utilizing combinatorial spaces of synthetically accessible molecules in our workflows. So we use extensively enamine real space, Chemspace, freedom space, and in later stages we come to so-called custom spaces, which could be built using so-called make-on-demand building blocks or custom-made building blocks. A few words about this synthetically accessible universe. As I mentioned, we widely used enamine X real space, which is over 5 trillion molecules up to date, and Chemspace freedom space, which is 142 billion molecules. So both of these spaces they're fragments, but they exist as a fragment spaces. And what is important that these spaces contain synthetically accessible molecules, which any molecule from this space can be synthesized with extremely high success rate, over 80%, within very reasonable period of time, 3-4 weeks for real space and a little bit longer for freedom space. I will walk you through the process and present our approaches which we utilize on each stage of the discovery process. So for hit identification, we have a number of tools, including high throughput screening, of course, virtual screening, but we also do, and we also work DNA encoded library approach, and we have special tools to accomplish fragment-based drug discovery. Our virtual screening platform includes a structure-based approach and also ligand-based approach. We have a number of libraries we could start with. They differ from each other by the size. Independent on the size, we could select the approach which we're going to use to virtually screen this library. Of course, it's classical molecular docking. We also widely utilize AI accelerated molecular docking, and of course, V-SYNTHES approach, which could help us to screen the spaces of the size of trillions of molecules. This is a virtual, our track record for our virtual platform. So on average, the success rate for our projects varies from 1.3% to 33%, and it's very much target dependent. We consider the hit molecules when we have the hit with activity less than 10 micromole. When it comes to hit expansion and SAR, so we have a number of approaches we can use to enable this stage. And of course, it's analog by catalog, and we have a number of search algorithms, how we could generate these analogs from chemically accessible spaces. We do 3D search, of course, as well, based on the shape or based on the pharmacophore of our hits. And we widely utilize a synthon search to enable and to design so-called SAR sets. So the idea here that our hit molecule is divided into the synthons, and then we could fix for example, 2 syntons and vary just one synthon. And it could get us a valuable information about structure-activity relationship. We widely utilized machine learning techniques to generate the follow-up molecules as well. When it comes to hit to lead and lead optimization, together with enamine, we enable the fast DMTA cycle. So the design could be done rationally by our med chemists, but we also can could utilize generative AI, in particular reinvent and optimize any of the properties of the molecules using this instrument. This is a project overview. So this is a project we have up and running right now. And for two of them, we kind of reach a lead optimization stage. And we really hope that in the nearest future, we could help our customers to develop as a preclinical candidates. I should mention that for almost half of these projects, we utilized the V-SYNTHES approach to enable hit identification stage. With that, I would like to conclude and give the stage to Stella.
Okay, but let me share my slide. Can you see it?
Almost.
Okay, great. Thank you very much, Olga, for a very generous introduction. It's really a pleasure, like always, to talk at Chemspace at Anamine. It's great pleasure to be part of this movement that you guys started probably like more than 20 years ago. Yuri should know more precisely, but like the first time we heard about Chemspace was about like 2007, and we tried to implement some fragment-based, some syntone-based approach. We chopped it into syntones ourselves and tried to order compounds, but it was like 20 million, the size of the whole space at that time. It was pretty expensive, so it didn't go to it. It had to wait for another 15 years, where like it really grown into great technology that drives a lot of pharmaceutical development this year. So okay, so with this. Today I will describe once again, for those who haven't heard, the what is V-SYNTHES, and touch on some new developments in the program itself and in their applications. So if I can move my slide. So okay, so this is the outline I just mentioned. So I will introduce V-SYNTHES and the applications to more and more challenging and clinically relevant targets, and describe like what's happening and where we are moving with the technology. Okay, so just to sort of get into what we are trying to do. We are trying to change the paradigm in drug discovery, where like usually it starts with just like finding a needle in a haystack using usually HTS or like simple virtual screening. So you get some hits, and then in the space that is already synthesized, described, it's not patentable. You need to do a lot of custom synthesis, moving to lead compounds and then drug candidates. So you're moving towards like along the in this overall like drug-like space, but it's not amenable to like parallel synthesis. It's custom synthesis. You need to order building blocks. You need to develop the synthetic routes. So it's expensive and long. So the idea is to switch to modular approach, where like the compounds are assembled like Lego, and this can be implemented at every step of drug discovery, from initial screening to like SAR by catalog optimization. And so the first hits that you get, they are in in this commercial on-demand space that was pioneered by Chemspace and Enamine. And when you move towards leads, you can stay in this space. You can expand it a little bit into tangible space with made building block that Olga just mentioned, and in sometimes like getting to like drug candidate without even doing medicinal chemistry. Usually you have to do like some medicinal chemistry, but again, it's relatively simple because the reactions are designed and optimized by Enamine, like to start with. So this should really democratize the drug discovery, at least the initial stages, and shorten the timeline from years to months. And hopefully get a bigger success rate because you can try more things like in early stages here. The early stages become like very accessible and can be done fast. So you can like really focus on optimization. The computational tool that we use here, we think of it as like yin and yang of drug discovery, physics-based and data-based, or deep learning or AI. So like on physics-based, we specifically use internal coordinate mechanics, where the molecules are present, like each atom of the molecule is presented with bond lengths, bond angles, and torsion angles in the molecular tree. And the molecular docking is so this internal coordinate mechanics presentation allows to speed up like all like energy-based simulations like 1,000-fold, often reaching conversions for many systems if they are well defined. Molecular docking is one of these well-defined systems where the ligand is presented in internal coordinates. Receptor is further simplified into the grid, potential grid representation, where all the like physics-based terms like electrostatic, hydrogen bonding, van der Waals, hydrophobicity, they are presented on as potentials on the grid. That is also softened to allow some flexibility. So this is like the docking algorithm. And of course, molecular dynamics is always a good tool to validate certain things and discover some interesting things. For example, this pocket in GPCR was found to accommodate a lipid that basically like walks into the pocket between helices. Pretty unusual move. So the ICM, of course, I wanted to thank the MolSoft team, Ruben Avagan, Maxim Tatrov, and Eugene Rausch, who works on this. For AI-based beta and data-based approaches, of course, we know there are Nobel prizes, not one but two, in the last couple of years for both the neural network technology itself and for protein design with neural networks and protein structure predictions like AlphaFold from David Baker, John Hopfield, and Geoffrey Hughes. So the machine learning, of course, the most important part here is data itself. So the data accumulated for ligand binding, for like in PDB for structures. Without PDB, the AlphaFold won't work. And then like the new technologies with like deep learning transformers, agentic AI and so on that are becoming increasingly important here. So one thing before I leave this slide, these two, of course, they are highly complementary. The AI is learning, or like deep learning is learning on the data generated by physics-based approaches. Physics-based approaches implement some new deep learning-based like scoring functions, for example. So there is a lot of crosstalk between two, and they're used like extensively together in like most applications. Okay, so my lab is developing some of those modeling tools and like for structure-based and data-based approaches. And initially we applied them to understand structure and function, to analysis of structure. So our favorite family, as Olga mentioned, is GPCRs. We like worked a lot with structural biologists and analysis of those, of how they function, what are the activation mechanism and so on. But most recently, we sort of started to pay more and more attention to applications of this knowledge to virtual screening and also rational design. I will talk today only on virtual screening, like in real space, but we publish a lot of like rational design, starting from known scaffolds, how to modify molecules to achieve new functions. Okay, so the virtual screening, the sort of breakthrough into ultra-large screening space started like 6 years ago with some work from Brian Scheuchert lab, where they show that switching from like about a million of few million initial molecules to hundreds of millions of molecules to screen, it allows to achieve like advanced compounds, high quality hit and leads much faster in weeks or months, because compared to years. So basically like size matters. You need more molecules to be able to get quality like starting points for drug discovery, and they tested it on several examples. So what are these ultra large spaces? Again, the drug-like space is enormous, 10 to 23 and more compounds. So when you like start with a million compounds, it's just a speck sort of in this space. And it is expected to give like a few hits, maybe. So if you identify hit and they like a few hits with relatively low potencies and probably not very not very favorable features for drugs. So once you got initial hit, there is a long way of optimization towards leads and drug, all in sort of general drug-like space without with custom synthesis. The paradigm changed with real space introduction by Chemspace and Enamine, where the like about 180 optimized reactions are applied to hundreds of thousands of building blocks, and this gives rise to like at that time 10 billion compounds, 30 billion compounds a bit later, which are virtual. You never synthesize the whole space, but they are real, so readily available in the sense that they synthesize in 3-4 weeks with 80% success rate. They maintain drug likeness and diversity. And again, as Olga mentioned, this is just the tip of the iceberg. There hundreds of millions of made on demand building blocks that can expand this tangible real space into like petascale and what 10 to 15, 10 to 18 compounds. Now, when you find their first hits like in this general space, of course, it also needs optimization, though usually like you get better hits just because of like size of the library. And the path to leads and to drugs, it lies through the same parallel synthesis. So within the same well-defined space that is easy to synthesize. Now, everybody understood that like going into these enormous spaces, now the bottleneck becomes the computing cost. So 30 billion compounds, like to dock them individually would take years, like on regular hardware, or like about $1 million, like the cheapest, the bottom price on like Google Cloud or Amazon Cloud. So the concept that we introduced is use the chemical modularity of real libraries. So if you have like a two-component reaction with two attachment points, R1, R2, and like 1,000 of R1 syntons, 1,000 of R2 syntons. If you enumerate them, it will be like 1 million compounds. But you can do another trick. You can enumerate only one side, find those partial molecules or fragments that fit into the pocket, again, partially, and then select the best, not one, but the best, like thousands of them, narrowing down the search, and then find the match for the second side, filling the pocket, and assembling the whole molecule. And this requires only like 2,000 dockings out instead of million. If it's three-component reactions, so it's like 1,000 times 1,000 times 1,000 billion on this side, and just 3,000 on this side. So the synton-based approach, it grows with the number of building blocks linearly, like arithmetically. The full enumeration, it grows geometrically. So it's like it really very fast goes out of control. So this is just a sort of an illustration and concept. The approach was implemented by the power couple in my lab, Anastasia and Arman Sadebekov, develop this virtual synton hierarchy of enumeration screening of V-SYNTHES. So basically, the idea is to assemble the molecules like inside the puzzle on the fly. And this in slightly more details. So you basically represent the whole chemical space of like reactions and syntons with minimal enumeration library that has all these syntons, that have all the reactions, but do not fully enumerate only one side of the molecule. You take this, call it MEL library, you dock them in your target, you select the best minimal fragments, usually like about 1,000, a few thousand out of 600,000, so already reducing the combinatorial space, and then enumerate them on the other side and like redock for the full molecule, so that for two-component reaction, if you have more three-component reactions, you do this like one more step. There are four-component reactions possible, so it's just like keeps enumerating until the molecule is full. The first sort of test, the whole thing is based on the whole concept is based on the idea that the binding of the initial fragments, mel fragments, it's then reproduced in the full molecules with certain accuracy. And we didn't have it in our first paper, but we checked it now in the paper that prepared to publication. So this is the initial fragment docked here, and this is the full molecule, and it I guess the examples shown here are too perfect. Usually it's like within 2 angstrom of MSD, but the poses of initial fragments, sometimes there are some small flips, small changes, but usually, and you can see in those distributions, it's under 2 angstrom difference between initial docking of the fragment and the full molecule. There, you see, like there's some distant bulges here that represent like when the whole molecule flips. It happens when there's like too much symmetry there, and like when the docking is not perfect. But usually the poses are reproduced pretty well. I should emphasize, not for all compounds, but for top ranking enumerated molecules, because like if the top ranking molecule that dock with high scores reliably dock, the compound that do not, the fragments that do not dock reliably, of course they jump around, they sort of flip. So but we are not interested in that, so we're looking into like really top compounds, and the higher like scoring compounds you get, the higher this sort of reproducibility is. So the second assessment is how relevant the scores of these fragments that you select. So we compared the sort of fully enumerated set after all these steps for selection through fragments to just randomly enumerated subset from the whole space, and we see that there is pretty high enrichment if you just use the scores, and we found that the poses also matter. So there are some non-productive poses when so this is an attachment point, and it's caught sort of in the corner of the pocket where it doesn't have space to grow. So if you attempt it to grow, the molecule will flip, and it loses like its sort of initial anchoring points. So if you find a way to reduce these cases of like non-productive poses, the enrichment gets to hundreds, to 150 for 2-component, 450 for 3-component reactions. In our original paper, the original paper, like in Science 3 years ago, on V-SYNTHES, it's also had full experimental validation. So we synthesized and tested 60 compounds predicted by V-SYNTHES, and 14 of them were sub-micromolar hits for cannabinoid receptor CB2. And then we done the first CR by catalog for the best 3 hits, and got from like 100 nanomolar hit to nanomolar hit to sub-nanomolar hit with also high selectivity to our target CB2. So the other validation was on ROC1 kinase, where we got the like nanomolar range, single-digit nanomolar range hits, like right off the bat from the screening. We didn't do optimization. Okay, so since then there was a lot of developments. So as Olga mentioned, we licensed the technology to Chemspace, and they are cranking quite a few projects. I won't be talking about them at all today. the V-SYNTHES program, again, it grows. There are some new deep learning components added. The size of the library grows, and we apply it to more and more targets. So this is like our collaborators within USC, like most of them at the medical school who come to us with their targets, and then like all over the country and outside as well. So we started the Center for New Technologies in Drug Discovery and Development, CNT3D, that basically like manages all these collaborations. We can now proudly show the like pipeline for CNT3D. You see, we haven't got to clinical trials yet, but we have a couple compounds in pretty deep into preclinical with like formulations being with the preclinical candidate selected and formulations being figured out. And then like other collaborations within USC and outside on other important targets. I think some of them are funded by NIH and hopefully will be funded in the future. One program is already licensed to the company. We started a company based on this A2R neuropathic pain, and there are other projects, like more than 15 now. So I do not show like at all some of the targets, but we have already some quality hits here. So our flagship sort of project that arose from ultra large screening from V-SYNTHES is 82 antagonist for neuropathic pain. I'm sorry. So it started with like barely submicromolar compound found in this GigaVLS, and the first SR by catalog got it to 100 nanomolars. And then we got NIH funding where it allowed us to synthesize compounds through collaborators. So it took like about 200 compounds to get to sub nanomolar, and then another 1,000 compound basically to optimize all the ADME properties and PK. So we now have a pretty solid preclinical candidate that shows high efficiency, high efficacy in neuropathic pain relief models in mice. So basically, the AMA401 is our old clinical candidate that shown efficacy in phase 2 clinical trials, but was discontinued because of off-target side effects. So our compound basically beats AMA401 in efficacy, getting practically to like normal sensitivity, restoring the normal baseline pain sensitivity in animals for a longer period. So it will be most likely once daily pill. It's orally available, and it has all the properties, and it passes through like first first PK studies in mice and rats. And so we continue this. This is now funded by new grant that just started recently, and we plan to go into clinic in the next couple of years. The second flagship project that got into preclinical development is CPLA antagonist for Alzheimer, and it targets an enzyme phospholipase 2, CPLA2 in ApoE4 pathway, which is strongly associated with early Alzheimer. This is enzyme. It's pretty challenging because the part of the pocket is flexible. We run screening with V-SYNTHES and got like 16% hit rate. This is initial screen. We got this already screened at lower concentration at 2 micromolar for the analogs on the like second round. And by now we got this compound BRF 50460 with sub nanomolar potency, which beats again the best compound that was available so far. This has quite a bit of like biochemical validation, but also it works in vivo. So it actually like in brain tissues, it protects against A-beta 42 induced synaptic loss in human tissue, brain tissue. So it's important that sort of it works in human brain, and it restores the it protects from synaptic loss in these experiments. Okay, so these were like project that like went very successfully, and the CPLA2 project basically it moved through all these stages in less than 2 years in academic environment. So just from like we do not have a grant for it, so it's like done on shoestring budget and pretty successfully. Now they're more challenging targets. So for example, TRPV is a transporter. It is well known. It plays critical role in neural development, cardiac function, and immunity in cancer. And it's an extremely hard target. So you know there are drugs for TRPV1, for TRPV2. There's basically no good compounds. But our collaborators in Vera Moisenko lab solved quite a few cryo-EM structures in different conformational states with compound that are like non-specific, but they bind and they show they open some cryptic pockets. So it's CBD bound somewhere like in the middle, piperlongumine, 2 APB and perbenecid. So like all over the place, the cryptic pockets that might work, might not. So it's quite highly flexible structure. Anastasia performed the V-SYNTHES screening for all 4 of these pockets, and so the first 2, the CBD and PL pockets, they had only 2 hits like between them. They are connected. That was the initial screen, but then we screened 2 APB side and found 4 hits, and then this PBC side finding 8 new hits. And again, we done it with different sizes of library. So the compounds were tested not only for binding but for functional activity. So like one of the best, 2 to 5. It turned out to be agopam. So it has some agonistic activity by itself, like shown here. But when combined with CBD, so you see this is electrophysiology already. So the CBD has signal which is greatly enhanced by addition of the compounds. And again, you can see like some activity from the compound itself, but not that much, and then highly enhanced CBD activity. They also solve the structure, though it's pretty low potency. We believe it should be somewhere like in 10 micromolar range, but they were able to solve the cryo-EM structure with our compound. I cannot reveal it because we don't have any IP for it yet. But it binds in this pretty well-defined pocket, and now we have like the structure that will help us guide the further optimization. So which is currently ongoing, and we're expanding the space to search for these analogs. Another challenging project is, again, it's transporter, which is considered to be like very challenging. It's GABA transporter 1 or GAT1, which is one of the SLC transporters family. And actually, the first discovered SLC transporter some 50 years ago. So it's a very important target that already has a compound, drug, tiagabine, that is approved anticonvulsant. It's not very clear how it acts because, like, even after the structure was solved, because it binds from extracellular side, but the structure was solved in the inward open conformation. It has some major side effects as well, because it's like low selectivity. So we done screening for two conformations, one of the like outward open pocket and the other for inward open pocket. And Jordi in my lab performed the screening. 131 compounds were purchased. 41 of them show some activity either as agonist or antagonist. And these are sort of the best compounds in 10 micromolar range, which is already very promising. Like, so it's a good start. We are optimizing them. So again, there is a whole range of like success or not success, or like very fast success towards like nanomolar compounds, or like more challenging targets with more challenges. One of the approaches, one of the projects, and like it's probably like extra challenging target that Chemspace, like Olga and Mikola performed for the cache challenge number 2, and which you probably know about this challenge that like more than 20 computational team competed. They overall more than like almost 2,000 compounds were tested for cache 2. And the target was so challenging that even the best hits are in like 10 to 100 micromolar range potency, even after optimization. What is interesting, the methods that people use, they're ranged from physics-based, machine learning, and crowdsourcing, and some combination of this. And the Chemspace performed pretty well with V-SYNTHES, finding this compound at 20 micromolar, which was also selected. And we're preparing a manuscript. Okay, so where do we go next with these technologies? So the just to show in scale how the so this is one of the versions of XReal that was used for screening in cache approach, 173 billion molecules. So, and as Olga mentioned, now there is a version of like almost 3 trillion compounds in XReal, which also improves some of the properties and slightly bigger molecule that's suitable for larger pockets. The again, the beauty here is that if you do like initial screening in like 1 million compound high throughput assay, it would cost you about 100K, bring no IP. But if you use like if you start screening with 3 trillion compounds of XReal, we like the cost of this project like be like tens of thousands of dollars, and it gives you unrestricted IP right off the bat, like for the scaffold that you discover. I mentioned we keep improving the program. So one relatively incremental step was V-SYNTHES 2.1, like improving automation of the post-selection algorithm. We called it CapSelect. So it builds for each mel fragment dock, starting from the second attachment point, it tries to build a chain of like spheres according to certain geometry rules. And if you cannot build any, so if it clashes with itself and the side of the pocket, it's blocked. If it goes outside of the pocket, it's sort of escape situation. And if you can build more than 2-3 spheres, it's more productive. So we have like a quantitative score that takes this into account and helps to select compounds. So again, this is some examples from the paper that we are preparing for publication. I don't know if you can see it, but the enrichment factors grow to like thousands for 3-component reaction. Okay, so this was 36 billion compounds. How to scale it from billions to trillions? And here we decided to use some deep learning, which is implemented like in the latest stage of like fully enumerated compounds. There are so many good compounds like being selected here that we want to screen like about 100 million like at this stage, which is like a bit too much for like individual docking. So what we do is we implemented like a convolutional network classifier that is trained on randomly. So from this 100 million, we select 500,000 compounds. We train the neural network model on this and deploy it to 100 million. It gives high recovery. It gives like another 200-fold speed up, which combined with V-SYNTHES speed ups gives like about 1,000,000-fold speed up compared to docking all the molecules. So there some validation of it. I probably don't have time for it. So just a few like philosophical slides at the end. So first of all, do we need trillions of compounds? Why bother? Like maybe like it's time to stop something. The answer is the bigger the better. And in this we are sort of we're in a good agreement with Brian Schoikert, who recently published the paper showing that even hit rates improved with library size. But first of all, like when you go from millions to billions, in million you just the number game is such that you can only find like 10 to 1 micromolar heats, and so there are like low chances of finding anything better. If you go to billions, you can find lead compounds, a lot of lead compounds to choose from, and even probably some compound that like directly can be drugs if you go into trillions. The hit rates improves the like the higher the more compounds with higher scores you can choose, the better the hit rates. So basically, the situation is that like out of a trillion molecules, with like 1 in million, even when 1 in million molecule is a good hit, usually it's like 1 in 10,000. You get like millions of potential candidates. So what if you can choose, if you can sort of narrow down to and corral sort of all million of hits there, what to do with them? You cannot synthesize million compounds. You cannot test them all. So how to exploit this sort of embarrassment of riches to get better leads and drug candidates? So first of all, like you want to improve hit rate. And for this, you can apply like several orthogonal scoring functions, especially like really orthogonal that like less dependent on specific structure conformation and specific like atomistic detail, for example, DL-based. There are even more rigorous but slow affinity predictors like absolute FEP. Now, just published paper on bolts 2, and we're playing with it to see how suitable it would be to like select to screen the last 100,000 compounds, for example, to test. There, you can apply additional filters for properties of the molecule, bioavailability, and so on. Blood-brain barrier, so you can cross check them with anti-targets for selectivity and so on. So if you have a lot of potential candidates, there's a lot of ways to improve selection. I mentioned deep learning-based RTCNN score implemented by MolSoft. It uses like pretty sophisticated combination of topological convolutions, like to represent the molecule itself, and radial convolutions to represent interactions in the pocket. It is trained importantly on clean PDB and generated decoys. So having negative examples in neural network training is super important. So this decoys thing, it changes situation. So it performs very well in benchmarks, and it can be used as an orthogonal filter for. So that's what we usually do. We have plot like scores versus RTCNN score, and select a compound that behave that have both scores good. Another important principle is you want to, and like advantage of structure-based is you can use known ligands to optimize receptor conformation. I won't go into this, it's pretty old publication when we used ligand-guided known ligands to guide the receptor conformation. Now it includes also waters in the pocket that are important for binding in many cases. Okay, one last philosophical slide. Where we stand, where this type of screening of aniline real space stands against generative models that are so popular. So V-SYNTHES, it takes target, validates it with known ligands, or like even optimize them, and do the screening in real space, getting real space compound for testing that are synthesized by enamine for a very reasonable price. If you the generative approaches, they sort of skip the validation state. They get the target. You basically, there is no way to validate structure in generative approaches. So you directly go into predictions based on ligands and the sequence of the target or structure predicted by AlphaFold, for example, and search for synthesizable compounds. So you predict synthesizability, some general synthesizability, analog and latent spaces. This gives quite, so it's probably it looks into pretty large spaces. The down and gives pretty like inventive compounds, but it downsides, it gives unrealistic binding poses very often. So the Postbuster established that like more than 80% poses are wrong. And it requires expensive custom synthesis like in there. So what where the field is moving in generative models is actually and what they used all the time is they used docking for validation and final selection. Sometimes like screening the last 100,000 molecules by Vina, which is probably not the best like way of doing things. And most importantly, there are quite a few recent papers where people working on generative approaches recognize that synthesis is really, so it can be done very fast, the generation of molecules, but synthesis is a critical step. And so there are several approaches published in the last couple of years how to constrain the generative approach to space, to specific synthon spaces like real space. So for example, SynFlowNet, and there are like dozens of approaches that sort of revert back to enamine real. So the whole thing, the whole idea of generative approaches was like based on idea that it will be like faster and cheaper and give more go more into like explore more of the drug-like space. But like this steps show that sort of it's not exactly the case. And in terms of like speed, V-SYNTHES per target is about like $300 for screen of computational time. But like the generative approaches are like about the same cost for GPU being spent. Okay, I will also wanted to say that there are different. We only discussed like hit identification and some optimization. There, it's still sort of yin and yang. But in for initial hit identification, physics-based approaches, in my view, still work better and like more straightforward sort of way of getting to result that are synthesizable and testable. There are other like starting points from known ligands, from pharmacophore, from known compounds that work slightly different, and like for fragment-based design also benefits from both physics-based and AI-based approaches. So it is really rapidly developing sort of nice competition and synergy between yin and yang here. So as a summary, modular V-SYNTHES makes possible fast screening of billions of compounds in real space, and with addition of deep learning, it can grow into terascale, trillions, and even more. The heats identified, this can be optimized in the same or even bigger combinatorial space. It provides cost-effective and fast entry points in drug discovery for challenging clinically relevant targets, and still the accurate structural information is critical, and we work and collaborate a lot with structural biologists. It's amazing progress that cryo-EM made in the last few years, giving a lot of detail that like AlphaFold bolts cannot predict yet. Okay, so with this, I would like to thank my lab and mostly Anastasius and Arman Sadebekov, who really critical in developing V-SYNTHES, and Tanin Nazarova, who continued this. Jordi, me, and Wujing, who contribute to development and apply the applications of these approaches. Our collaborators at the USC and across the country, and most importantly, Chemspace and Enamin and Olga, Kola and Yuri at who are really pushing the boundaries and the edge of science here, like in terms of chemistry, and also with collaborate. Okay, thank you. And yeah, by the way, we do have positions, so please apply both as students and postdocs. Thank you.
Thank you very much for the amazing talk. Yeah, I enjoy like each your talk, and yeah, it was amazing. Thank you. And we have a question right now. So, will V-SYNTHES work for 4-component reaction?
I'm sorry. What about 4-component?
Will V-SYNTHES work for 4-component reaction?
Yes, we actually had like some examples like from the very beginning. Right now, there's not that many 4-component reaction in Enamin, but the combinatorics works in the way that like 4-component reactions would be actually the sweet spot for V-SYNTHES, because of combinatoric, there will be like relatively few building blocks that so very small MEL library, very fast to dock, and then you just run not like 2 rounds of enumeration, but like 4, and you get like full compounds. We have better enrichments going from 2 components to 3 components, so I would expect even higher enrichment numbers for 4 components. Okay. Yeah, we hope, and I mean will actually bring more of these compounds. Yeah.
Yeah, I think it's something that that could happen, of course, but yeah, talking about like 4-component reactions, it may result quite in quite large molecules, like eventually, and yeah, it may not be suitable for every single target, but at the same time, we do have a demand for larger molecules, like beyond 2 or 5. Yeah. Okay, the next question. Do you see the limit in the size of the chemical space that V-SYNTHES can handle?
Well, with we see that like it's very far on the horizon. The so we now test it, and like it works very well with like C-trillion version of XReal. So it's 10 to 12. We see that it can be very effective with 10 to 15. And again, it's always compromise. So you, of course, you start losing some hits like when you go into high space, and it's not. It's just starting from trillions. It's already impossible to handle all potential hits. It's like millions of them. What you do with that? Uh, so, but initial screening can be very easily done, like beyond 10 to 15.
Okay, so there are like a few questions. First of all, how to apply for postdoc position in your lab?
Just drop me an email.
Okay. Okay, in 82R Agonist project, what does it mean, the catalog SAR?
Well, SAR by catalog, sort of, it's some idiom sort of that people use. So basically, like to get into more details, we do synthon-based search. So we have hits, and we search for analogs using usually expanding from the initial screen space to XReal, um, uh, and to do synthon-based search, and then we usually sort of one of the um, tricks we usually do is we explore just like very small variations. So some sort of like medicinal chemistry approach, like one atom here, one like heteroatom replacement there. So sort of like within Tanimoto distances, like 0.1. To find like really close analogs. And then the other complementary sort of approach that is to like have a broader search, or like more exploratory, like swapping whole syntones, like focusing on known syntones, and like performing docking, or performing some like deep learning based ligand based sort of changes.
Okay, thank you. We have two last questions. Are there any tips to prioritize the molecules after RT-CNN scoring and docking scoring, if there are still many kind of hits?
Well, again, it's all in ranking. We have like some empirical sort of threshold, what is like a good dock molecule for this target. It varies from the target to target, but we like if there are known hits, if there are known like sub-micromolar compounds, at least like in Campbell, we by docking those compounds, we can establish sort of the expectation for where should be like good molecules. So and if we apply like these thresholds, even very strict ones, we usually have like tens of thousands of compounds after V-SYNTHES and RTCNN both applied. So this last leg, where one really wants to have binding affinity predictions with we estimate that like our score predictions, they reflect binding affinity plus minus 2-3 orders of magnitude. So like we can predict that it's nanomolar compound, it will be micromolar compound. Okay, that still works, right? But we want to, like, if we want to further sort of narrow the search in meaningful way, we need predictors that give you affinity predictions like with like plus minus one order of magnitude. And there are claims that ABFEP can do this. In our hands, like what we see, it's more than like 2 orders of magnitude, but still some improvement from scores, and it's very slow. So it is really like a bottleneck now. This sort of accurate predictions of the like for final selection that would probably if you really develop such an approach that like generalizable to many targets, it can be very useful and allow to like get to this golden like 100 compounds for like 200 compounds for synthesis with like really higher hit rates, high success rate.
Yeah, exactly. Okay, the last one question. How would you suggest to select the number of enumerated compounds to be docked? The default is 2.5 million in the script, but this would affect the number of symptoms being filtered.
Whoa.
Well,
The number the number of fragments for enumeration, right? So basically it is flexible number. The parameter sets the number of total molecules to be enumerated, and so it goes like from the top of the table and enumerate this. Well, again, it's the more the better. It should be defined by throughput of your like cluster, and so like usually we target and the whole point of like adding deep learning accelerator into this was to keep the number of docked molecules to like under 1, maybe 2 millions, so that you never have to like dock more than that, never have to go beyond like day or two of like cluster use.
yeah. Okay, I think we're done with questions. Sarah, thank you so much for your time, for very, very nice talk, and for Q&A session. It's always a pleasure talking to you, listen to you, and yeah, I hope that we will continue our working together in the future.
Thank you very much, Olga, and thanks everybody for your attention. That I overstayed a little bit, but thanks a lot.
No worries. Thank you. Have a great day. Bye.